dear colleagues and future colleagues. I'm Gábor Nyiri from the Institute of Experimental Medicine. It's a great pleasure to be here and to talk to you about work regarding the brainstem control fear and its memories. Actually, I started my research just about 200 meters away in an old building of the Szeged University. At that time, I mostly worked in hippocampus and synapses. But today I would like to talk to you about more the behavioral aspect of brain research. So the brainstem is, of course, participates in several, in a wide range of different functions, including the modulation of pain, awareness, consciousness, autonomic functions, and also provides the brain with several modulatory transmitters, neurotransmitters, dopamine, serotonin, norepinephrine, relaxin. So for this reason, for a long time, we thought that the brainstem probably just a modulatory part of the brain. But now we know that it is not really the case. The brainstem, although it's a very ancient part of the brain, can actually very directly and quickly influence higher order brain functions like memory and fear processing of negative experience. So today I would like to talk to you about some previously unrecognized and unrecognized cell types and cell populations in the brainstem that are so important for fear memory processing that their inhibition itself can impair the negative experience processing, while at the same time their activation can actually induce fear in itself. So of course, fear processing in the brain is processed in many different areas of the brain. You know, the brain, what the brain has to do? It has to avoid negative experience and try to explore as much positive experience as possible. So of course, most of the brain deals with these kind of problems and issues. So, but coping with negative experience is important because the animal quickly needs to recognize the situation, needs to understand that something bad is happening, have to react to it and to make memories about these experiences so that later they can remember and predict these negative experiences. So coping with negative experience requires a system that actually understands whether the thing that happens is better or worse than the animal expected. The negative valence circuitry that recognizes the situation. For this process, we need the lateral habenula that is very important in deciding whether the experience is negative or not. It also provides the negative experience, helps to predict negative experiences, activates the medial part of the ventral tegmental area, dopaminergic neurons that target the prefrontal cortex. So this system is important in understanding whether the experience is negative or not. Now, coping with negative experience also requires the septohippocampal system, these nuclei in the brain that are important in acquiring memories of these events so that the animal will be able to predict these events in the future. This requires the parvalbumin cells in the medial septum that are pacemaker neurons. These help the hippocampus, that is the primary memory center in the brain, to promote the theta rhythm in the hippocampus, and it is important because the theta rhythm helps the hippocampus to explore the environment and to make new memories. But of course, there are other regions of the brain that are important in memory formation for the negative experience. This is mostly a contextual memory formation part of the brain where the animal needs to understand where it is and what's happening. While, for example, the amygdala is important in acute fear memories, like because the amygdala has a very strong connection with the auditory cortexes. So when the animal hears something that an auditory cue that makes him remember something bad in the past, then this amygdala helps to the animal to remember. This is the acute fears. And of course, there are the bad nucleus of the stria terminalis that helps to provide the long-term, to process the long-term effects of negative experiences. So it's responsible mostly for the anxiety. But for a long time, we didn't really know how these brain centers are actually coordinated when something bad is happening to the animal. So we knew that the brainstem has an ancient part called median raphe region. And this region has been known to regulate mood, fear, and anxiety. However, those cells that were discovered in this median raphe region could not really explain why this area is so important in these processes. We knew that there are so-called vesicular glutamate transporter 3 positive cells here. These are kind of glutamatergic excitatory cells and serotonergic cells and GABAergic cells, but didn't know which of these cells do the important job. So several years ago, we decided to map the cellular architecture and transmitter phenotypes of this area. And we, of course, could detect the somatostatin neurons and the vesicular glutamate transporter 3 positive neurons, but, and a lot of GABAergic cells, mostly local cells. But we realized that a quarter of the neurons are unknown here. So we were lucky and found that these cells are actually vesicular glutamate transporter 2 positive neurons. These are also excitatory glutamatergic cells. And when we filled these cells up, we realized that these neurons give the largest output of the median raphe. So for these experiments, probably not all of you know these, but nowadays we have certain types of transgenic mice that help us to investigate certain populations of cells in the brain. And the Vigil2-Cree mouse line is one of them. In these mice, we can actually label specifically a specific population of cell that express Vigil2. And we use a special virus that is only expressed in these cells. So we could actually label these median raphe neurons, these new neurons, and look at where they arborize in the brain. And we found that they specifically target the lateral habenula and the medial part of the ventral tegmental area, exactly the areas that I talked about. Exactly those centers that are so important in evaluating the negative valence of the events. So I won't be go into details, but we also looked at whether they are really glutamatergic excitatory cells in the median raphe. So we made in vitro slice preparations and recorded from postsynaptic cells, and we could easily see that they are excitatory neurons, and all their postsynaptic currents could be completely blocked by glutamatergic receptor blockers. So these are excitatory neurons. So what do they do and how they work? We wanted to know whether there is any kind of feedback between these nuclei. We knew that the lateral habenula, that is so important in this process, actually lateral habenula is also very important in depression. That's been discovered not too long ago, and there was a long history in the science that it's very important in that. So the lateral habenula gives feedback to the median raphe, but whether it gives feedback to these cells was also a question. So what we did is that we labeled the lateral habenula neurons with red viruses and the median raphe neurons with green viruses, and we could see that the red cells were surrounded by green, the green cells were surrounded by red terminals. So they actually contact each other. This could be very important, because in the initial phase of a negative experience, the animals very quickly has to recognize that something bad is happening. So the neuronal network has to be up and boosted. So this is probably the best way to do it, because the activation, one cell activates the other that activates the original cell, so there is a positive feedback loop. Now, of course, it has to be stopped somehow, and this is where probably these GABAergic cells come in. As I already mentioned, like almost more than 60% of the cells are GABAergic in the median office, so they probably make sure that these cells are stopped in time. So what do these cells actually do in the brain? So we put down an silicon probe, it's so-called silicon probe. It's an electrical device which you can use to record from these cells. There is a method called optical tagging that I don't have time to explain to you, but let me just say that we can actually select these cells and make sure that we record only from them, from these VGUT2 cells. And we realized that when we presented the mice with a negative experience, an air puff that they really hate, then these cells started to get activated very much. If we presented just a mild negative experience, like a strong LED light, they were mildly activated. But when we gave reward, these cells didn't care. So it really seems like they really selectively respond to aversive negative stimuli. Okay, so far so good. But what do these cells do if they are activated? So can we activate them? There is a method called optogenetics. Some of you may have heard about it. With this method, we can actually selectively activate a certain population of neuron in the brain with light. So this is what we did. We put an optic fiber above these cells that were labeled, and we put the mice into a cage where they could actually choose where they would like to stay. But on one side of the cage, we started to illuminate these cells. Now, of course, in case of the control mice didn't care because their cells were not sensitive to light. So when they entered this part of the cage where the light was illuminating, entered into their brain, they didn't care. It was just a normal situation for them. But when we used another mice where these cells were very much activated by light, the mice couldn't dare to enter that part of the box, because they felt something very bad is happening. They, of course, didn't know what's happening, but it was very, very bad. Now, this suggests that these cells signal a very bad experience for the mice that they really don't want. So this is what I'm showing here, this experiment that I showed you on the video, that they have a real-time place aversion. So immediately they hate the firing of these cells. But in addition, we put the mice back into the same cages, and the next day, it was clear that the animals remembered. So they really hated that part of the cage, so they tried to avoid it. It was a significant effect again. Now, the question is whether the mice thought that something bad is happening inside them. What is responsible for this? Is there something from outside? So we tried to put the mice into a social environment and activated two groups of mice. I mean, these mice were litter mates. They brought up together from the same moms and dads. And we activated some of them together. So I will show you. We put them together in the same cage, but the control mice, when we put together these control mice, they didn't care. Of course, they were litter mates. They were friends. But if we used a chemogenetic tool, in this case not light, but a close-up in N-oxide, it's a drug, that were injected into these mice. They were injected in the control mice as well, but they did not have these channels that reacted to these chemogenetic tools. So when we put together these mice where these VGUT2 cells were activated, they started to attack each other. These graphs show that they were very aggressive at the time they were put together. So it seems that if you activate these cells, the animals feel that something bad is happening to them, so they try to do something about it. And if they see someone in their cage, they think that other mouse was the reason. So they attack it. And also, this is what happens when we put a little intruder, a little other mice, harmless mice into their cage. They also attack it. So, okay, so far so good. So it seems that this is very important in activating the negative experience circuitry. But the question is whether it can activate the memories for the animals. Can it help to form memories? So it seems that it has something to do with memories as well. Because we realized that these new cell types also send a lot of fibers to the medial septum and the vertical limb of the diagonal band of Broca, this part. And these are exactly the cells that I told you that are important in activating the hippocampal theta rhythm. So we saw that they surround specifically the parabolic cells in the medial septum. They really try to activate them. And they are indeed the parabolic cells that target the hippocampus, because we could label them back from the hippocampus if we injected dyes into the hippocampus. So these are really the cells that make sure that the hippocampus is active enough to form memories. And actually, this graph just shows, I won't be able to go into details, but this just shows that there is a significant elevation of theta rhythm in the hippocampus. So this really prepares the hippocampus to form memories. Now, of course, we wanted to know if this is just happens to help memory formation, or this is really necessary to form negative experience in the animals. So we tried to block it when the animals needed it. So this is a little bit more complicated experiments, but I hope I will be able to tell you how we did it. So this is an auditory signal. So the animals hear a voice, a tone. And then at the end, we shock it a little bit. We surprise it with a mild food shock. They are a bit frightened and don't know what's happening. So they associate the noise, the tone, with this negative experience. But at the same time, when they receive this frightening food shock, we illuminated their heads. So we had, of course, two groups of mice. One, the control, which did not have the right channels, so they didn't care about any kind of illumination. So, of course, they behave like they should behave, because they show freezing behavior. They just stop, and they seem to be very much afraid of what's happening, because the next day when we put them back into the same cage, they remember that they've been here the day before. So it's really frightening. But those mice in which we inhibited these very same cells, they could hardly remember that anything happened to them in this cage. So they couldn't really process the negative experience properly. And then in the next day, we put the mice into a completely new environment. They have never been here before. And generally, in this case, if a mouse has experienced some negative events, then they generally show some generalized fear behavior. So they're a little bit afraid anywhere. And indeed, these control mice were a little bit afraid, but the mice that were blocked were not. And then when we showed an auditory cue to them, then the animals, of course, the control mice showed a strong fear behavior, but the animals which were blocked here, at least partially, showed much less, significantly less fear. So what does it mean? It means that these cells are not only sufficient, but also necessary for a normal processing of the negative experience. Now, of course, the last question is whether these cells, where do these cells receive their inputs? We can actually test that using so-called monosynaptic rabies technologies. I also don't have time to explain how it works, but just simply say that we can inject into the cells some viruses that can go into the contacts, the synaptic contacts, and go back one synapses and labels those cells that gave synapses onto them. And with these methods, we could actually test that there are many different areas of the brain that mostly are important in negative experience processing that gave synapses onto these glutamatergic neurons. So these cells are really very much integrated into the whole circuitry and are very important in negative experience processing. So the take-home message is that there are still several cells and cell populations in the brain that you have to discover, because we don't even know that they exist. And there are several areas of the brain that we still haven't discovered yet. There are new nuclei and little part of the brain that no one knows is there. So I'm closing here, and I'm of course very thankful to my group, the hardworking students. Some of them started their career while they were in the secondary school. So far before actually they started their university years. And some of them are already very successful scientists. And of course I am grateful to the to other collaborators, because it's a teamwork. Thank you very much.
Thank you, Gábor, for this really exciting lecture. And now please stay for the prize.
Before a very important announcement before we close this session, that at 3 o'clock this afternoon, there is a very important point in our program. And this will be the naming and foundation stone laying ceremony, which is not here, but at Pacsirta utca 23, which is the corner of Kossuth Lajos Sugár and Pacsirta Street. It's a 20-minute comfortable walk from the city center, and we really, really would like to ask all the students or the teachers, guests, to be present on this occasion. So thank you very much, and now we would like to give the certificate and close this session.